let us continue further discussion. Hello everybody. So in the previous lecture we were looking at the processes of transport. I told you about the uniport mechanism, where you have got movement of the ion in a particular direction. You can have the symport mechanism, where the cation and the anion can move the same direction, and you can have the cation and the anion motion in opposite direction and referred to as an antipode process. Now these are extremely important processes in biological systems. For example, we are aware of the fact that we need to maintain the sodium and the potassium ion concentration in our body at the optimal concentration, so that the body functions in an optimal way. And what is the most important requirement in the body is we do work, and when we do work, the system burns energy, and the energy is released by the conversion of the process of ATP to adenosine diphosphate and then to adenosine monophosphate. So there is a stepwise process, because ultimately we need to convert glucose to carbon dioxide and water. So in this process what happens is that whatever the energy is stored in the body, now this energy has to be released. For example, when muscle contraction takes place, the reactions start where the ATP has to be converted to ADP, and this process of release of energy is catalyzed by a class of enzymes called ATPases, of which the sodium and potassium ion ATPase is the most important example. Also, we need to keep in mind that 1 mole of ATP releases 35 kilojoule per mole of energy. So we essentially now have the ribose part along with the adenine part, and then this is covalently connected here. As you can see, I am just drawing a part of it. So this is covalently connected to CH2O, PO minus, P double bond O, O, P double bond O, O minus, O, P, O minus, double bond O, O minus. So we have this 1, 2, 3, 1, 2, 3, and then 1, 2, 3. So we have this adenosine triphosphate, and now there is a cleavage of this particular bond, which is going to give us, and this happens in the presence of magnesium ion, and the delta G of this process is minus 35 kilojoule per mole, and it gives you the H2 PO4 minus ion plus the ribose, adenine and ADP, and this is the inorganic phosphate which is released. And magnesium acts as a catalyst for this particular process, and so it is a very important process, because the PO bonds are the strongest bonds, and when they break, maximum amount of energy is being released when it converts into the inorganic phosphate and the adenosine diphosphate. And now coming to the application of this, we first need to know the biochemical concentrations of potassium ion and sodium ion in millimole per kg of the body. So in the this is the human intracellular fluid. The potassium ion is 92, the sodium ion is 11, whereas in the human extracellular fluid, which we also call as the blood plasma, it is 5 and this is 152. Now, if you look at this process here, we can show it in the form of a box here. So, what happens is that now inside the Ai concentration is greater than Ko plus, and sodium plus outside is greater than sodium plus inside. So, this is the so because of this, what happens is that the natural concentration gradient is in a way that the potassium ion flows from higher concentration to lower concentration. So, it goes from inside the cell to outside of the cell where the concentration is lower, whereas for the sodium ion it goes from the outside to the inside of the cell. But what we need to do actually is that in this biochemical processes, we need uptake of potassium ion. So, potassium ion has to be taken inside now, potassium ion has to be taken in, potassium ion has to go inside, and this is against the concentration gradient, and the sodium ion has to come outside. So, this has to be excluded and this has to be included inside. So, and this is against the concentration gradient, and this process of pumping potassium ions from the outside to the inside of the cell and sodium ions from the inside to the outside of the cell is done using this Na plus K plus ATPase enzyme, because this is not a natural process, it is occurring against the concentration gradient. And what this enzyme does is that it essentially scavenges the sodium ion, from outside, from inside and puts it outside, and potassium ion it takes from outside and puts it inside, and this is against the concentration gradient, and this is done by this ATPase enzyme. And in the biological system, in the human system, this process of transportation of potassium ion against the concentration gradient and sodium ion also is done by a class of molecules which is referred to as ionophores, and this is done using a carrier mechanism. So, there are different types of mechanism which can transport the ions, and one is the carrier mechanism and the other is a channel mechanism, where there exists channel like structures at the bilayer across which the ions can be transported, whereas in a carrier mechanism the ions which are actually hydrophilic will now have to be transported across the bilayer, which actually is of a lipophilic nature, and because it is of a lipophilic nature, we will have to modify the environment around the cation so that the cation can be transported effectively across the bilayer. So, in this regard, the carrier mechanism has to operate, and the species which will be able to carry these potassium ions across the bilayers are referred to as ionophores. And traditionally, the traditional examples of these naturally occurring ionophores are nonactin and valinomycin. And just to look at the structures, these are pretty large structures actually, but just to look at the structures, I can draw this for you. So, the structures are as follows. So, this is 3 times. So, here we have got DHYI, this is called D-valine, this is lactic acid, and that is L-lactic acid, which we call as L-lac, this we call as D-val, and this is dihydroxy isovaleric acid, that is DH5I, and we have got the D-valine as well, and then this becomes D-L-valine. So we have DH5I, D-valine, L-lactic acid and L-valine. Now these essentially make up the valinomycin structure, and here you can see that you have got the polar parts and you also have got the non-polar parts. So when you have this kind of a arrangement, this kind of a very specific spherical arrangement, so we see that the ionophore is also pre-organized because this is the cyclic structure, and when what happens is that when it interacts with the potassium cation, now because the ionophore has to capture the potassium cation, it will now interact with this oxophilic, it will now interact with the oxophilic or the hydrophilic, it will interact with the oxophilic or the hydrophilic carbonyl groups. Okay, and now when these oxygen atoms will be pointing towards the potassium cation, it will be encapsulating them, and now the isopropyl groups will be presenting the isopropyl groups shall present an lipophilic exterior. They will now present a lipophilic exterior, and because of this what will happen is that now the potassium ions can be effectively transported across the bilayers, and let us see how this takes place. So to understand this ion transport mechanism, chemists actually now started making synthetic ionophores, and in this regard the 18-cron-6, which we have already discussed, functions as a very efficient ionophore. So in this regard we would like to understand the ion transport mechanism. So let us look at this process. So we now have the chambers here. So we have this as the lipophilic membrane, this is the hydrophobic part, and we know that the hydrophilic part is always solvated by the water molecules which are in the extracellular space or in the intracellular space, the water molecules. So this is the extracellular space, this is the intracellular space. So we now have the potassium ions, which now, so we say we have the potassium ions, and these are actually first captured by the organic ligand, which we call as the ionophore. Okay, and where will they be captured? Essentially, when they will be captured by the ionophore, then only they will be able to be transported across this lipophilic membrane. So before we go there, let us first take the metal ions, which are actually solvated by the water molecules. So this is the aqueous. So this is the aqueous layer. Aqueous layer. Now what happens? The desolvation has to take place. This is the solvent loss, and now the metal ions, they will be captured here. Okay, they will be effectively, you will have the oxygen sites here, and so the ionophore comes and it picks up the metal ion and it forms this particular complex. Then this particular complex will be transported from the outside to the inside of the cell, and it will now present the lipophilic sheet towards the interior of the cell, and it will present the metal ion to the hydrophilic part, and will be now released into the intracellular space. So we have the metal ion, it will be transported by the ionophore, which exists in a particular conformation. On encapsulation, it it has this particular arrangement. It is now being transported and then released into the intracellular space, where now you have the solvent molecules which will pick up the metal ion and get solvated. So, this is how the metal ions are being transported across the bilayer. This is to show you that synthetic equivalents of the naturally occurring ionophores have been designed, which can transport the potassium ion very effectively, and this is referred to as a carrier mechanism. And naturally, as I told you, this particular process is being done by NAT plus K plus ATPase enzyme. This is the particular enzyme that catalyzes this particular process, and as we have now realized that in the case of the natural system, what is happening is that the potassium ions are being taken from the inside. The potassium ions will have to be taken inside from the outside against the concentration gradient. Okay, we can show this thing to you. So, here we can see that the potassium ions have to be taken from the outside to the inside, and the sodium ions have to be taken from the inside to the outside against the concentration gradient, and the overall reaction which takes place in the natural system is as follows. So, we have 3 sodium ion intracellular plus 2 potassium ion extracellular plus ATP 4 minus plus water in the presence of magnesium, taking 3 sodium ion to the extracellular plus 2 potassium ion to the intracellular, plus now releasing ADP 3 minus plus HPO4 2 minus plus H plus. Okay. So, we have got a net charge of +1 here and net charge of +1 here. So, we have, you see, the conversion from ATP to ADP, and we also have the ion pump mechanism which is operating, where the potassium ion is being taken from the outside to the inside, and this is against the concentration gradient, and sodium ion from the inside to the outside, and this is against the concentration gradient. So, we have this movement of these ions across this particular bilayer, which is actually facilitated by the sodium plus K plus ATPase enzyme, and this actually enzyme, to tell you, is a membrane protein. So, this is a membrane protein which is having molecular weight of 294 kilodalton, and the active site actually consists of 2 peptides which we call as alpha beta, and overall this alpha beta dimer makes up the active site, and this is what this enzyme, if you look at the structure, it facilitates the transportation of the potassium ion, and this has also been explained overall. This particular transport process has also been explained in the way of a pump storage model. So, you have a process where you have if you plot the energy as a function of the spatial coordinate, you will see that we are actually pumping the ions, we are actually pumping the potassium ions and the sodium ions against the concentration gradient. So, this is the pumping mechanism, which is actually the active process, and we also have got the passive process, the gating mechanism, which is the passive process. Okay, so, the natural mechanism, as we saw, was the concentration of potassium ion inside is greater than that of outside. So the natural mechanism is to go from inside to outside, but we actually need the reverse process. We need potassium ion to go inside, and this is done using a pump mechanism. And in this case, we will say that the metal ions are pumped from low concentration to high concentration against the concentration gradient until a dynamic non-equilibrium state is reached. So, in which active pumping is balanced by diffusion, and in order to activate the gating process, a certain any hormone or chemical messenger, for example acetylcholine, can activate and open up selectively the gated ion channels allowing the passive and therefore rapid flow of ions back to equilibrium, resulting in flow of current. Okay, so this is the overall explanation, that metal ions are being pumped from low to high concentration against the concentration gradient, because you have a dynamic non-equilibrium state, and in which active pumping has now to be balanced by diffusion, and the process of diffusion will start only when you have a stress to the system, or you have an impulse, or there is a chemical messenger or a hormone that can activate or open up these gated ion channels, which now allow the rapid diffusion of the ions back to equilibrium, and this process creates a flow of current and plays an important role in nerve transduction. That is how the nerves carry the electrical current, is by this process. So, this signal nerve transduction in the nervous system is done in this particular process, and we also realize that overall this is of relevance in the biological system, and this is how the sodium ion and potassium ion together make the operate as a pump mechanism, and to maintain the equilibrium in the body. And so with this we now will just take up another example, where we will be looking at another very important application of these cations. So we have looked at the sodium potassium pump. We can also look at another example, where just like you have got these naturally occurring ionophores, people were also interested to look at other species in which cations have to be taken up for performing the necessary biological processes at the cellular level. And another very important class of molecules are referred to as siderophores. And in case of siderophores, what has been observed is that the essential element which has to be taken up is iron, and iron, as we know, has got enormous applications, because it is plays a very important role in oxygen uptake, and hence is of relevance in cellular processes. And the problem with iron is that iron 3 is insoluble. For example, it exists as rust in aqueous solution, and at physiological pH of about 7.4, the solubility of hexaquarine 3 plus is around 10 to minus 18 dm mole per dm cube, whereas in case of microorganisms the optimal concentration is 10 to the power 7 minus 7 mole dm cube. So, how do we achieve this required concentration of iron 3 plus inside the microorganisms? So, we need certain ligands in the biological system which will capture Fe3 plus and deliver it to the cell, and these kind of highly effective iron complexing agents are called siderophores. They come from the Greek word meaning iron bearer. So, the siderophores were discovered in as early as 1911, but it was only in 1951 that the first natural siderophore was discovered, and which was called as mycobactin. Mycobactin was the first natural siderophore which was discovered, and after that there was another siderophore which is called enterobactin, which has been discovered as well. And what is essential is in this particular complexes or in this particular ligand, these are pretty large size ligands, but what they have is that they have got the catechol moiety, and in this catechol moiety they have the dihydroxylated groups, and these dihydroxylated groups get deprotonated at slightly alkaline pH, and then because they are O minus, they are able to now interact with the iron 3 plus and capture it very effectively. So, what essentially happens here is you have now got, in case of enterobactin, you will have this CONH backbone which is here, and then what will, I am giving you a skeleton view of this. What is the most important part is that you will now have this O minus, O minus, O minus, O minus, and then you will have the third one from here. So, you will have O minus, O minus. This is how they will come out, and these will now, this entire set of 6 oxygens will now capture Fe3 plus, forming a trigonal prismatic geometry. Okay, and so we have got these deprotonated hydroxyl groups, and so these catecholate moieties are there. The catecholate moieties are there. The overall charge on the ligand is 6 minus, and then it interacts with iron 3 plus, so it forms Fel 3 minus. This is the overall charge on the ligand now, and the binding constant is very high. 10 to the power 52 is the binding constant for, and so these are extremely stable chelating agents for iron, and this is what makes them very efficient siderophores, because they can capture iron 3 plus. The binding is extremely efficient, and it is able to form the trigonal prismatic geometry where iron 3 plus coordinates with these deprotonated hydroxyl groups to form the FeO6 3 minus species. So, thus we see that nature has developed very facile and very robust mechanisms, so that these processes go on in a very efficient manner over millions of times, and very efficient processes. And supramolecular chemists have tried to design ligands which can actually mimic the active site of the naturally occurring substances, and this is what also constitutes an important application of supramolecular chemistry in biology, that people now started designing more better and better molecules which can actually mimic the natural processes which are occurring in the nature. For example, people want to now develop oxygen uptake models, for example, or make models which mimic the plant photosynthesis processes, and so on and so forth. So with this, I hope you have been able to appreciate the role of ion transport in biological systems, and in the next set of lectures we will now switch to other systems which particularly involving self assembly processes. Thank you.